Namaste to all students. Myself JV and Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about the Genomic Library. This session is related to recombinant DNA technology course of program BTech FBT 4 So basically, what are the steps of genomic library constructions? So in the previous session, we have already discussed that what is the gene libraries and what are the two types of genomic gene libraries. First one is the genomic library and second one is the cDNA library. So in this session, we are going to discuss about the genomic libraries. So what are the steps which can be followed in the genomic library construction? So first of all, isolation of the DNA from the cells, digestion into small fragments, introduction into suitable vectors, insertion into bacteria, which is transformation process, and further isolation of the recombinant vector by and then collection of the genomic library. So all these steps are very important for the construction of genomic library. So first of all, we will discuss about step 1 in detail, which is isolation of the DNA and purification. In eukaryotes, we will prepare the cell nuclei, remove the proteins, lipids and other unwanted macromolecules by protease digestion and phase extraction method. But in the case of prokaryotes, we will do for the extraction of the DNA directly from the cells. And further, we will go for the digestion into small fragments steps. In this step, we can use the physical sharing method, in which we can do the pipetting and mixing. Second procedure we can follow, or alternative procedure we can follow, which is restriction enzyme digestion. It is partial digestion which is preferred to get a greater length of the DNA fragments. Then we go for the introduction of this digested small fragment into the suitable vector. So each fragment is very different and have a unique DNA sequence. So we will go for the insertion of that DNA fragment into suitable vectors, including plasmids and bacteriophage vectors. Vectors are digested with the same restriction enzyme as we have used already in the gene of interest for creating the sticky ends. So we will use that same restriction enzyme for digestion of the vectors and have to create sticky ends. Then we will go for the ligation process. DNA ligase enzyme we will use, and we will ligate the recombinant, we will ligate the vector and digested vector and the digested gene of interest for the ligation process. The resulting molecule is basically recombinant vector which is having gene of interest. Then in the introduction of gene of interest into suitable vector, we can get the resulting molecules which are recombinant vectors. Further, we will go for this recombinant vector insertion or transformation into the bacteria. So we insert the recombinant vector into the host bacteria, which can be E. coli, and generally we use the competent cells, and strain is most commonly used is DH5 alpha. The microbes can be grown in the culture which is having this recombinant vector, and they are made to take up the DNA. They replicate their genome along with the vector genome contained with them, produce clone of the original genome, and this collection of the clones, which contains all the sequences found in the original source, including the sequence of interest, forms the genomic library. For understanding it more, we can use the diagrammatic representation by which we can understand the whole process, that how the multiplication and the production of the clones done. And in the independent plasma replication process, we completely know that in the independent plasma replication, these plasmids will be replicated within the cell. On the other hand, when we use the host cell replication process, we can get so much colonies of the cells, each containing copies of the same recombinant plasmid. And single cell is also doing the independent plasmid replication. On the other hand, these host cells are going to replicate and creating the more numbers of the cells which are having these recombinant vectors, and these recombinant vectors will self replicate or independent replicate within the cells. So that's how we can get the large number of the cells, and we can get the large number of the recombinant vectors or recombinant plasmids, and this is how we can do the multiplication and production of the clones. And one more thing we can understand by the practical uh, handling, that first of all, for the genomic library construction, we have to do the isolation of the DNA from the cells. We can simply see that in the eukaryotic cell there is well defined nucleus and which is having the chromosome, so we will isolate the DNA from the cell. This is the genomic DNA. Then on the other hand, we will prepare our plasmid vector which are suitable for the cloning method. Then we will go for the digestion with the same restriction enzyme for the genomic DNA and for the plasmid vector. That's how we will get the cleaved DNA with the sticky ends. These are the sticky ends. And on the other hand, we will get the cleaved vector also with sticky ends. Then we go for the ligation process with DNA ligase enzyme, which create the ligation in between recombinant vector as well as in the gene of interest. So further, we will get the final product which is recombinant vector. These recombinant vector will be transformed into the competent cells, and now we will get the multiple clones of the cells which are having the plasmid vector. Now these are the living kind of a storage of our gene of interest. Then we can go for the DNA isolation or plasmid isolation, and we get the collection of the genomic DNA, and that's how we can create a proper storage, or we can create the genomic library by the following all these steps by the sequence. So that's how we can create the genomic library. And what are the steps involved in the construction of the genomic library? In our previous slide, we have already discussed, but here we are discussing in the detail. So, to create a human genome library, a researcher begins by extracting and purifying DNA from human cell. The purified DNA consists of extremely long strands. To begin working with DNA, the strands must first to be cut into manageable size. The DNA therefore is digested with restriction enzymes which cut the DNA at specific sequences. The restriction enzyme cut the DNA into thousands of smaller fragments, each of which may contain one or more genes. Each fragment is different and have unique DNA sequence. To create the library, each fragment must be inserted into loops of DNA called plasmids. A plasmid is a type of vector that allows the DNA to be shuttled in bacteria. The plasmids are digested with the restriction enzymes and then sealed to human DNA using DNA ligase enzyme. The resulting molecules are the recombinant vectors. The recombinant DNA molecules are added to the bacteria, and bacteria are made to take up the DNA. When bacteria have taken up the DNA, the entire collection of the cells and DNA represents a human genomic library. The recombinant plasmids remain separate from the bacteria's own DNA. They will not be mixed, or they will not be ligated with each other. So that's why it is making the easy to extract again. Why what are the use of the genomic library? Why we use the genomic library for the um, storage of the genomes or DNA? So researcher can explore the genome of an organism to learn more about the genomic structure and function. They can map the genome, identifying the locations of specific genes, helps to develop tests which can be used to locate genetic variations including mutations. Useful in recombinant DNA technology, helps to generally genetically modify organisms and produce clones of desired type. Genomic library construction is the first step in any DNA sequencing projects. Genomic library helps in identification of novel pharmaceutically important genes. So that's why we are very much fond for the genomic library because there are so much uses, there are so much importance of the genomic library, and that's how we need to create, we need to develop a genomic library at a large scale. So this is the sum up of this session. If we go for what are the important points and what are the important key notes in the context of a competitive exam. So first of all, you have to remember that what are the steps of the genomic library constructions. So first step is isolation of the DNA from cells. Second step, digestion into the small fragments. Third step is introduction into suitable vectors. Fourth one is the insertion into bacteria. Fifth is DNA isolation, and sixth one is the collection of the genomic DNA library. So, and one more thing, like if you go for the diagrammatic representation of the genomic library construction, you will understand that the same restriction enzyme will be used for the digestion of the genomic library as well as genomic DNA as well as for the plasmid vector. So you have to give answer yes. We have to use same restriction enzyme so that they will create the same complementary sticky ends, and it will help for the ligation process. It will easily ligate with each other. And one more thing, what is the terminology for insertion of the recombinant vector into the plasma, into the bacterial cell? So the process is known as transformation. And in the transformation process, generally scientists or researcher use the heat shock method. First of all, you have to use the vial of the bacterial cell into the uh, into a optimum temperature for a optimum seconds. After the incubation period of the heat, we have to introduce a recombinant vector into the that process along with the bacterial cells. And after getting the heat shock, bacterial cell will be loose up, and their pore will create becomes large, and then these recombinant vector will introduce into the bacterial cell. After completion of the specific optimum duration of the heat process, we have to immediately transfer that vial into the ice, just because of a temperature difference and temperature down process. That bacterial cell will seal up again and their pore size will become less again. So that's why the recombinant vector is not able to come back or come outside, just because cell has been packed. So that is the principle of the heat shock method for the transformation process. First of all, we have to give heat process, and then for a specific time period, and after giving the heat process, we have to shift it into the ice process. Just because of a temperature large difference in the heat process, cell become loose and pore size will become large, so that the recombinant vector easily can enter into the cell. And when we transfer that vial into the ice process, so icing process, so cell pore size becomes less, and you can simply say that recombinant vectors can be sealed within the cell. So that is the concept, and that is the principle of the transformation process. And after the transformation process, we can go for the multiplication of that recombinant cells, and after the multiplication of the recombinant cells into the suitable nutrient media, we will collect the cells and we will isolate the DNA from these recombinant cells, and that's how we can isolate the DNA, and we can store that DNA, and we can create the genomic libraries. So that is the main concept of the genomic library construction, and these all are the basic steps which can be followed during the genomic library construction. So that's how this is sum up of this session, and this session is powered by digital version 2.0, Jyoti Vidyapeeth Women's University, Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.